Welcome, everybody. Welcome to London Futurists. My name's David Wood. Today we're going to be looking at the future of healthcare and medicine. And it's good news in a way that every time we look at the BBC News or some of the other news sources, there seems to be a flood of articles about breakthrough techniques and therapies. So we read about the mechanisms for improving people's immune systems by reprogramming the immune systems to strengthen them. This seems to have a lot of promise for dealing with various kinds of cancer. We read about remarkable ideas with stem cells that can be used in lots of different kinds of regenerative medicines. We read about 3D printing of organs or replacements for parts of the organs in the body. So there's a lot of news. But it's very hard often to know what to make of all of this. It's hard to know which of these stories are grounded in strong research, which of them are still vague ideas, which of them might apply to mice that have been specially genetically altered in the first place to make them more susceptible to a disease. It's also hard to know how far these treatments are through the process of turning them from a research idea into real practical medical therapies. But somebody who does keep on top of all of these questions is Professor Stephen Minger, and it's my great pleasure to welcome him here today. So let's give a round of applause to Professor Stephen Minger.
So good afternoon, everyone. I hope you found it as hard as I did to come inside today, because it's a really lovely day outside, so I'm really grateful that you gave up the sunshine to come sit in the dark in here and listen to me pontificate about advances in healthcare. And I'm really grateful to David for the invite. I think the last time I spoke at a big event like this, a futurist event, was 3 years ago, right after I transitioned out of GE as global head of R&D and chief scientist. And at that time, my talk was really more kind of basically geared toward stem cell research, and we never got to the really interesting stuff beyond that, which is today I'm determined to do, even if we have to sit here until 6 p.m. So, and we can do this however you want. And feel free to ask me questions as we go on. If I'm not making sense, yell at me and say, can you make that a little bit more discernible, please? And if you have questions, we can either take them at the end or during my presentation, whatever you feel like doing. So I have my own independent consultancy company now, as I'll point out to you in this talk. I moved to the UK in 1995. I'm British, as you can tell by the accent. I'm soon to be Slovenian, as I've had enough of Brexit and am relocating to Ljubljana as of this week. I will keep my company and a lot of the science that I do here in London, but I'm actually going to move to Ljubljana. So dober dan. So I was at King's for about 13 or 14 years. I headed up a big stem cell group. I'll show you. I've gone back, maybe it's part of the, sorting through your stuff over the last 20 years and trying to decide what to keep and what to get rid of. And in the process of doing that, I came across a whole bunch of old stuff I hadn't thought about for a long time. So this talk will be somewhat historical. I'll show you some of the things that we did when I was back at King's that I think are still relevant today for where healthcare is going and where research in healthcare is going. But in 2009, I was acquired by GE Healthcare, one of the largest healthcare organizations in the world, or at least they used to be. I'm not sure that they are doing very well at the moment, those of you who follow the markets. But I'll show you what we were trying to do at GE. I was global head of R&D, had about 15 groups worldwide, about 500 people. And then the last couple of years that I was there, up until 2015, I was chief scientist, which meant that I was responsible for their long-term kind of strategy. Where should healthcare go if you're sitting there in 2015? What should GE invest in that's going to be big in 2025 or even 2045? And that's the bit I really want to get to at the end. And so now I'm just an independent private citizen. I consult for a number of biotech companies, for a number of investors and venture capital funds who ask me to do due diligence on science that they feel like I have some expertise in. And I'm happy to talk about that if that's of interest to you, if you want. Okay, let's rock. So, my background is in neurodegenerative conditions like Alzheimer's and Parkinson's. I did my PhD in the late 80s at Albert Einstein College of Medicine in New York with probably one of the leading Alzheimer's researchers at that time, a Welsh guy named Peter Davies. And one of the things that really grew frustrating as I was doing my PhD in 88, 89 was the fact that we just had no tools to really understand anything about Alzheimer's disease. So for those of you who are knowledgeable about this, we didn't have mice, for example, that became Alzheimer or displayed Alzheimer pathology like we have now. We didn't have transgenic mice. We didn't have knockout mice that had any relevance to the disease. And experimental animals and primates do not get Alzheimer's disease. Okay, this is a specifically human condition. And so I became ever increasingly frustrated because all we had really was post-mortem brain tissue. And you can imagine, looking at somebody who's had Alzheimer's disease for 30 years down the microscope tells you shit all about what causes the disease or how it progresses. You just see the end stage of the disease. And rarely would you get somebody in the very early stages of disease who maybe had a heart attack. So most of the material we had were really late stage disease, and it's just very intellectually dissatisfying and frustrating. And so I was having a long talk with my boss about this, and I discovered actually that maybe I didn't need to understand how the disease was caused. Maybe I didn't need to understand the etiology of Alzheimer's disease. But maybe I could do something that could impact on the disease without really understanding what was happening, okay? And where that came from was actually a series of studies in Sweden, in a little small university town in Sweden called Lund. Any of you know where Lund is? By Malmö. It's across used to be quite a long trip from Copenhagen, but then they built a really nice bridge, so now you can get from Copenhagen to Lund in about 2 hours, okay? Really nice little small university town. And a really group of pioneering neuroscientists and experimentalists discovered that you might be able to repair the brain in Parkinson's disease, okay? So let me show you how this works. Parkinson's disease is, again, a human disease. Animals do not get Parkinson's disease. Primates do not get Parkinson's disease. Most of you can lose 70% of the neurons that sit down here, that project up here, that make a substance called dopamine. You will, and I will, naturally, during the course of aging, lose 50, 60, 70% of these cells, and we will have no symptoms of Parkinson's disease at all. If, on the other hand, you lose more than 70%, you will start to get Parkinson's disease, okay? Parkinson's disease was described back in the 1810s by an eminent British neurologist named Parkinson, hence Parkinson's disease. Parkinson's disease, unlike Alzheimer's disease, is predominantly, if not exclusively, a motor disease. And it's characterized by what's called bradykinesia, which is really slow movements. So Parkinson's patients become very rigid. Just for a patient with quite severe Parkinson's disease, stand up may take 2 minutes. And then when they walk, they tend to have problems getting going, and then they shuffle. And then they freeze, and then they shuffle. Okay? Initiating movement is really, really difficult for them, and keeping movement going is really difficult. And the second thing is generally they have really pronounced tremor. Sometimes worse on one side than the other, sometimes bilaterally they have tremor. Okay? There's a drug called levodopa that one of my professors at Einstein, Oliver Sacks, if you've ever seen the movie Awakenings, Oliver discovered levodopa as a drug for patients with, in that case, catatonic Parkinson's who'd had this part of their brain fried during an influenza crisis that hit America. But Parkinson's alleviates the motor symptoms. So in the early stages of Parkinson's disease, if you take levodopa, you move. Your tremor disappears to some extent, although it's usually kind of like this. You may have tremor in your fingers or you may have tremors in your mouth, but you can move. But the drug has on-off effects like this, so you have to take it all the time. When it starts to come off, you start to get slow and rigid again, okay? It does nothing to halt the progress of the disease, and over time you have to take more and more levodopa. And you end up having really quite severe on and off phases. And in the on phase, you go from being able to move a little bit to having all of a sudden really writhing excessive movements like this, which is called tardive dyskinesia. Okay? As this progresses, the drug becomes more and more ineffective, and you become more and more rigid and rigid and rigid, and ultimately you basically die of the disease. Okay? So it's, except for people who have Parkinson's combined with other neurodegenerative conditions like Alzheimer's, which probably 20% will, it's pretty exclusively the loss of these cells down here, which this is through the midbrain, so down here. And these cells actually are darkly pigmented, which is why this region is called the substantia nigra, the black substance. Okay? These cells actually have melatonin in them for some reason, which we don't really understand. It probably has to do with dopamine, because melatonin's partially in that pathway. This is about an 80-year-old person who's lost quite a few of these cells, but does not have Parkinson's disease. This is the equivalent brain section through somebody who does, of the same age. You can see huge amount of atrophy, complete destruction of these cells. They're completely gone. Okay? So Anders Bjorklund, Rusty Gage, Steve Dunnett, Mark Pashansky, Ole Lindvall began in the late 60s and 70s to begin to think about trying to replace these cells to see if that would work. Would that provide therapeutic benefit? So first they tried cells from, and this was all done in mice and rats. First they tried taking these cells from adult mice and rats and putting them into adult mice and rats that you can lesion to create Parkinson's disease. They don't naturally get Parkinson's disease, but you can destroy these cells in rats and mice with a toxin. Okay? If I take tissue from an adult mouse, put into an adult brain, it dies immediately. The tissue does not survive. So adult to adult is not going to work. Then they thought, well, what if we take young mice? Young mice did not work either. What if you take really young mice, like had just been born? Doesn't work. The cells do not survive transplantation. What if you take fetal mice? That is, mice that have not been born, which are still in the mum, and take them out. And what they discovered was there was a critical time in mouse development where you could take the equivalent of these cells in a developing fetus, and if you transplanted it up here where these cells could engage with their target neurons, you could basically alleviate the symptoms of Parkinson's disease in mice using fetal transplants. Okay? Now again, this is a chemical lesion. This is not mice who naturally get Parkinson's disease. It's a chemical lesion where you wipe out these cells, and I'm now giving you back new cells. Starting in about 1985, after a few thousand mouse experiments, they went to humans. They did no primate studies whatsoever. And basically, by staging human fetuses to the equivalent age of mouse fetuses, they found that if they transplanted tissue from the same brain region that I showed you, so from human aborted embryos, human aborted fetuses, if they microdissected this brain region down here, from a 6 to 7 week old human conceptus, which is 1 centimeter head to toe, and transplanted that up here in the brains of Parkinson's patients, they could get substantial clinical improvement. And this is like 1985, 1988. Okay? So they published on this. This paper is actually from '99, but this is a man who had been followed at this point in time about 10 or 12 years. He's now been followed close to 30 years. And this guy's very interesting. I think he's patient number 4 in the series. So if you just focus on this side of the screen, this is imaging using a technique called positron emission tomography, or PET. And what it does is it measures the amount of dopamine, the substance that these cells make, in his brain. Okay? So what you see here is, this is the normal range. If I'm imaging, this would be you guys and me up here, okay? This is how much dopamine our brain makes and uses. We're in the normal range. This guy, prior to transplantation, you can see he's already way down. He's only making about 40%, okay? And it's pretty equal on both sides. And here's what the scans look like. It's just basically color-coded from the amount of dopamine that he makes. This guy was quite unusual, despite the fact that it looks like he's got bilateral Parkinson's disease. One side was more affected than the other. And so the choice was made to transplant him only on one side because he was one of the first patients, patient number 4. He gets transplanted with human fetal tissue, and slowly but surely, over a period of about 3 years, his transplants take him back up into the normal range. Okay? Now, he's under immunosuppression because, as I'll tell you in a moment, he got tissue from a number of different embryos, all again 6 to 7 weeks of age. In utero, not 6 to 7 weeks after birth. This is 6 to 7 weeks in the uterus. Okay? Again, you're this big. Around here, he's taken off of immunosuppression, and our big concern was when you remove immunosuppression, his immune system would recognize the fetal tissue as foreign and it would take it out. That didn't happen. You can see even after removal of immunosuppressive drugs, he's still up in the normal range. Even more critically, this guy's brain is still degenerating. The transplants do nothing to interfere with the disease process. And you can see what's happening in the side that didn't get transplanted. He continues to regress. And basically 10 years later, he's basically got no dopaminergic function on the other side of his brain. This guy is now 30 years post-transplantation, roughly. I think he was transplanted in 1986, 87, somewhere in there. Still alive. Once immunosuppression was taken away, he came off of all of his drugs, and he's never gone back on drugs. So in essence, the transplants not only stabilize the disease, but as the brain continues to degenerate, the grafts actually improve functionally. They get better and better and better at compensating for the fact that his brain is degenerating. Now, if we did this 30 years ago, why the hell are we not doing this now? Particularly in this country where we have incredibly good guide. I mean, part of the reason why I came to the UK was to be able to do this kind of work, because you can't do this in the US. Here we have incredibly good guidelines that have been in place since at least 1990, called the Poking Horn Guidelines, which allow for individuals who are undergoing elective terminations to donate their embryos, their tissue from the embryos, for research and for clinical use. Okay? So not everybody would want to do that, but it's certainly available. Why don't we transplant more people with human fetal tissue? A, how many 6-week-old fetuses am I going to get access to? I need minimally 4 to 6 per hemisphere to treat one patient. That means I've got to find 12 6-week human conceptuses from individuals who are undergoing elective terminations, and I'm not allowed to interfere with the process of how these terminations are done, which in the old days was basically, without being too crude, they were suction abortions. They were basically aspirations were under ultrasound. The gynecologist would go in with a Hoover and just literally suck it out. And you would end up with fragmented tissue. What was brain, what was, the rest of the embryo was impossible. And again, it's 1 centimeter. Logistically, this is a nightmare. And we've only transplanted about 200 people worldwide in the last 30 years. But this works. This is brain transplantation. Everybody thinks this is 50 or 100 years off in the future. This works, and it's worked 30 years ago. We just don't have the right cells. Okay? So the point is, when this was done, the only other cell transplantation that we had was bone marrow transplantation and organ transplants. This isn't a stem cell transplant, but it sure is a cellular transplant. It's dissociated brain tissue that's injected in numerous needle tracts with multiple deposits in a very discrete and very targeted region of the brain. Remember, again, we're transplanting the cells not throughout the whole brain. We're putting them into a very small almond-shaped structure. Okay? And we're not asking the cells to regrow these really long pathways, which is impossible in the adult brain. We're just asking the cells to grow little neurites and synapses on their target cells. So it takes about a year and a half for you to see improvement in these patients, but when it happens, it's basically lifelong. The patients who haven't improved did not get enough tissue. I don't know if any of you have known Jeremy Thorpe, infamous Tory MP. I got to meet Jeremy a couple of times. Jeremy had transplants, but he only got transplanted from 2 embryos, and he got no clinical benefit whatsoever. You need minimally 4, probably 6, ideally 8 per hemisphere. You're just never going to do this. So the hope is, and what I started to work on when I went to, so after I saw this, I said, okay, I don't need to understand how Parkinson's disease happens. I don't need to understand how Alzheimer's disease happens. I just need to be able to apply transplantation technology to this. So I went to UCSD in La Jolla to work with a guy named Fred Rusty Gage, and Rusty had been one of the guys in Lund for 5 years, had set up his new lab in California, and I was one of the first people to join him. And the idea was, could we take tissue from an individual 6-week-old fetus and figure out how we could turn that into an equivalent of 1,000 fetuses worth of tissue? So in 1992, when I joined Rusty's lab, we didn't know anything about stem cells. Nobody even used the term. We talked about cells in the bone marrow that we use for bone marrow transplantation, where you take a patient with leukemia, you destroy their own bone marrow because they have leukemia, and that's where it comes from, and we're now going to give them cells back from somebody else. Okay? But we didn't even know about stem cells to any great extent. So went into Rusty's lab. The idea was, let's take tissue from a 6-week-old fetus. Again, we had to do all this work in mice and rats because we were not allowed, thanks to Ronnie Reagan, to work on human tissue. So we were trying all sorts of things. We were trying to immortalize the cells basically with genes that cause cancer. So we were sort of creating cancer neurons, or cancer precursor neurons, that we could expand, and we thought maybe that would work. And in the process of doing that, what we discovered that there were actually cells in the brain at about 5 weeks that we could take out of the brain and expand. That is, I could take a single 5-week-old equivalent embryo, isolate cells from the midbrain, from the forebrain, from the hippocampus, wherever, put them into a dish, and under the appropriate growth factor conditions, I could expand them to 100 times more. So our lab in 1992-93 discovered that there were stem cells in the developing brain. And without exaggerating, I think this really kick-started the stem cell revolution, because it was like, well, shit, if you can make stem cells from the brain, maybe you can make stem cells from the liver, maybe you can make stem cells from the heart, maybe you can make stem cells from the kidney. So for many years, from about 92-3 onwards, there were lots of us who were digging around in fetuses looking for cells that had not yet become a fully formed tissue, but we knew that the progenitor cells had to be there. Now, doing this in humans is impossible. If you think it's hard to get access to 6-week-old human abortions, how would you like to get a 3- or 4-week abortion? I mean, how many? I've never been pregnant. Some of you have. Did you know you were pregnant at 3 weeks? And if you did you have a termination? And remember, this is before the progesterone drugs, the RU drugs, which you could maybe get access to 3- and 4-week-old embryos. So this became really a roadblock for all of us, and I gave up. By 1995-6, when I moved here, we had tried to get access to human fetal tissue here in London. I went around to every major termination clinic in London, talked to all the eminent guys and ladies and said, could I get access to 5-6-week-old human abortions and tissue? And they said, you're dreaming. No way. Maybe one a year. So what was our alternative? I know that I can't take cells from anything past this because they're too mature. They don't survive the implantation procedure. So you have to go the other way. About that time, Martin Evans, a good friend, I'll show you a picture of him at the end. Martin, in 1981, together with Gail Johnson, had showed that in the mouse, you could isolate a population of cells that when you put them in the dish, you could expand them to huge numbers, and you could take a single cell, play with it, and you could put it back into an embryo, and you could make a mouse that now expressed your gene. This was the advent of designer mice. This was mice who expressed genes for Parkinson's, for genetic forms of diseases like Parkinson's, Alzheimer's, Tay-Sachs, cystic fibrosis. You could, in essence, take any human gene, put it into a mouse, and you could make a humanized mouse. Okay? And this is revolution. I mean, if you go up to the Welcome in Hingston, they are taking all 26,000 human genes and they're making individual mice strains that express each of those genes in mice. So you will have 26,000 strains of mice overexpressing human genes that they normally don't express. Okay, so this technology, Martin won the Nobel Prize in 2007, and it's not surprising because it's just revolutionized, experimental work. So we started thinking, well, okay, so these cells come from and I'll show you a bit more about this in a moment, but in essence, these cells come from the earliest state of development. And if they make a mouse, that means they have to make every other tissue. If they're competent to make a whole mouse, they have to be able to make everything. And all of a sudden, we could make neurons, we could make liver cells, blah, 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 blah. We're having a field day with this. This is great. And mice cells grow really fast. And you can differentiate them really quickly. And so we were really excited when in 1998, a guy named Jamie Thompson in Wisconsin for the first time showed he could do this in humans. Whoops. I'll get to that in a second, okay? Because this became, for us, a big watershed moment, okay? So people are working on mouse cells here. We've sort of given up here. But, there are still cells down here. Despite the fact I said you can't use these in brain transplantation, you can use them for other transplants, okay? So most of the field over the last 30 years has been, in essence, focused predominantly on these cells that come from the bone marrow. Initially, this was the major source. But now, ever increasingly, people work on the equivalent cells that come from cord blood, from the umbilicus of a baby that's just been born. So basically, the baby has in its cord and in the placenta the equivalent of what you and I have in bone marrow. It just has less of it. But that's where the baby's vasculature comes from at the beginning, okay? And once it's born, its ability to make its own blood cells is derived from cells that initially are in the cord, and then they migrate into the bone marrow, and now the baby can make its own blood. But these cells are pretty amazing. It's shown here. This is kind of a schematic of what the bone marrow itself looks like. And the cells fall into really two major populations. One are hematopoietic stem cells, or blood stem cells, and these cells give rise to all the blood lineages. So every day you make new white blood cells, new red blood cells, new immune cells, new T cells, new B cells. And you do this from the day you're born until the day you die. Somebody calculated that every second you make 3 million new red blood cells your entire life. 3 million a second. Think about the metabolic demand on your body to produce that many cells every second of your life. And that's just your red blood cells. What about the other cells in your body, okay? So these cells are crucially important. If you get leukemia, either in one of your lymphoid cells or so these B cells are B cell tumors, lymphomas, and T lymphocyte tumors. If these cells go out of control, basically up until very recently, your only way to avoid dying from this was to have this whole system completely destroyed and to get a transplant from a related donor or somebody else who was a good immunological match. Right? These other cells that sit in the bone marrow are what we call stromal stem cells or mesenchymal stem cells or MSCs. And they have two functions. One in vivo is really to control the production of these guys. So you don't get too few, you don't get too many. So they're kind of the master control cells. But if I take them out of bone marrow or out of the cord and put them into culture, they make bone and they make cartilage. They also make fat. Fat's not very useful, but making bone and cartilage is. Okay? If you go into a website called clinicaltrials.gov, which I did yesterday for a friend of mine who's looking for a gene therapy trial, and you type stem cells into clinicaltrials.gov, this is the website in the US that all American clinical researchers who want to go to a clinical trial, they have to register their trial on that website. It's an absolute requirement in America. Most European clinicians will also put their trials there, and ever increasingly a lot of people outside of the US and Europe are. Have fun. Yesterday it was 6,300 6,371 trials. 6,371 stem cell trials, of which about 90% will encompass these two cell types. Okay? These guys down here, we're trying to make bone, we're trying to make cartilage. I work with the clinic in Ljubljana where people who have had bad teeth and have had most of their teeth taken out, you tend to lose the bone in your jaw. And so we're actually taking MSCs, turning them into osteoblasts, and basically growing new bone that is size specific for their mouth. Okay? And there's a number of us doing this. There's a group in Israel called Bonus as well. There's another set of cells here that make cartilage, and so people are making cartilage for knee repair, and my friend Richard Arefo is using these cells to make hip, and so there's a lot of interest in regenerative medicine using these cells. Okay, so even though you don't have stem cells in most of your tissues as adults, these are still really, really important. I've already told you about the ability to make red blood cells. You have cells in your gut that every day have to replenish the endothelial lining of your intestine because it's the food and everything that you eat goes through it, it just shears it off. So you have to rebuild your intestine every day. Okay? What about your skin? Do an experiment. Go out and buy some black sheets. I thought this would be cool to have black sheets. It's not very cool at all. Wearing black clothes is probably okay because you can shake them out every day, but get some black sheets and you'll realize how much skin you lose every day. I mean, it's like we're just like giant dandruff balls. I mean, it's really quite amazing. What about hair? I mean, we all some of us brush our hair every day, and there's always hair that we pull out, but yet our hair keeps growing. So again, one of my friends estimated that if you live to be about 80, there are probably 8 of you standing next to each other of all the cells that you've ever made in your life that replenish all these tissues that we grow every day. But if your immune system attacks your pancreas, you don't make new insulin-producing cells, and you have to go on insulin replacement for type 1 diabetes, right? Your liver, I can take any of you who don't spend as much time as I do in the pub, I can take your liver and I can go in and I can whack out 3/4 of it and it will grow back. If on the other hand, people who spend more time than me in the pub, if your liver gets fibrotic and sclerotic, it will not regenerate no matter what you do. So it's something really weird about liver we don't understand. In healthy organisms, you can replenish it really easily. If you get type C hepatitis, if you get NASH, which is a really nasty non-alcoholic, basically liver fibrotic disease, your liver does not regenerate. And if you get cirrhosis, you're screwed. All you can do is get an organ transplant. Why we have such regenerative capability in one clinical setting but not in another, we don't understand at all. The pancreas has no regenerative capability whatsoever, nor are there stem cells in the adult pancreas that we can take out, engineer, and put back in people who have type 1 diabetes. Okay? So we have some stem cell populations which are really, really crucial in keeping us alive. We have other tissues where they're just absolutely gone, probably at birth. Okay? And that creates a problem because if, for example, like me, you want to create new islets for transplantation in patients with type 1 diabetes, I have no sources of cells from a fetus or anything else that's going to give me islets. Okay? Ever increasingly important, I think, and this is looking pretty far ahead, you actually have stem cells in your brain. Not only the ones that I was taking out of embryos, at 6 weeks of gestational age, but again, as we discovered in Rusty's lab back in the 90s, adults have stem cells in their brain that give rise to new neurons every day. Okay? First, we discovered this in mice. And in mice, in rats, it's pretty interesting. Most of the cells are found, this is in human, but they're found in what's called the subventricular zone around the ventricles. And these cells make new neurons that migrate into the mouse's nose every day. Because mice use their olfactory system quite exhaustively. Neurons get exposed to pathogens, dust, chemicals, and so these neurons get killed because they're exposed to the air. So the mice have to replenish them every day. But it also turned out that the mice have a few cells in a very high real estate area of the brain called the hippocampus. They make new neurons every day. And this system is susceptible to environmental and chemical manipulation. Okay? So this has all been done in mice and rats. But basically, there's a whole bunch of things that if you give them to mice and rats, the number of new cells that they make in their hippocampus, the really important ones which are involved in learning and memory, go way up. Sometimes as much as 3 or 4-fold. Okay? There's a whole bunch of things, on the other hand, that if you do to mice and rats, the number that they make goes down 2 to 3-fold. Okay? So a lot of this work we did in California. When I joined Rusty's lab, we were 25 people, and when I left to move here in 1995, we were almost 100 because this was so hot. Okay? So look at the good things. Running. If you take a mouse and you give it a running wheel, and it runs, the more number of new neurons it makes every day. And that's true in humans, we think. Caloric restriction. If a mouse eats 40% less, it lives twice as long, and the number of new neurons goes way up. Pregnancy is biphasic. So when the mice gets pregnant, they make more and more new neurons. After pregnancy, the number of new neurons goes down and almost completely disappears. And this we know is true in humans as well, except for by imaging. So brain volume in women postpartum is about 30% less than it was before. And it takes about a year for that to come back. We don't understand that biologically, I have to say. Statins. A lot of us think statins should be put in drinking water. Anti-cardiovascular effects, CNS effects, et cetera, they appear to be kind of wonder drugs. Aging is probably the biggest factor here. So this is what we knew in 1993-5. Rusty, together with a colleague, Peter Erickson in Gothenburg, who unfortunately had passed away really young at 49, about 10 years ago or so. Peter and Rusty showed that in the adult human brain, this is also true. Now, it's almost impossible to do this kind of work in human brain. How can I tell whether you're making new neurons today, tomorrow, next week? I can't image them using PET like I showed you in the dopamine stuff. I can't use MRI, okay? So I have no ability to measure what's going on. Peter came across a group of patients that were being treated at Karolinska who had really advanced throat and mouth cancers. And these patients are really difficult to image structurally because their tumors are embedded up into their larynx, the palate, et cetera. So what the medics were trying to do was gauge how quickly their tumors were growing in response to treatment, but imaging wasn't providing them with the information that they needed. So they relied on what we had used in experimental animals, and that is they injected them with a drug called bromodioxyuridine, or BRDU. And BRDU is non-toxic at a certain dose. It can become toxic. And basically what happens is BRDU gets taken up into any cell and incorporated into newly created DNA. So think back to biology. Every time a cell divides, it has to rebuild its chromosomes, right? Because when you have a cell that has one set of chromosomes, 2 pairs of each, when that cell divides, both sets of chromosomes go into the daughter cell. So you have to reproduce both sets of chromosomes. If you're a cell that's dividing and I give you bromodioxyuridine, it will get taken up and put into the chromosome. And we have an antibody against bromodioxyuridine. You can see it here. So all of these little dark cells that you see are cells that at the time this drug was administered to these cancer patients, their brains were making new cells. Isn't this cool? Now, the downside of this was the only way that we could see these cells was when the patients came to autopsy. Okay? But what this definitively showed, and I'm not showing you the other data, is just like rats and mice, you have 2 brain regions. This is the lining of the ventricle, and this is in the hippocampus, where we make new neurons every day. Now, those of you who are paying really, really good attention to me and just listening to every word, you might make 100 neurons after this talk. Those of you kind of snoozing in the back, or in the front even, you might only make 5. Okay? So remember, this system can be manipulated. So in an enriched environment, I have to say spending time with David Wood counts as an enriched environment. Would you not agree? And later at the pub, it will become even more enriched.
Excuse me, is nitric oxide at both sides, or is it a different phenomenon?
So this is mostly inflammatory. I'm not an NO maven, so it's biphasic. This has really to do with inflammation.
Okay.
My big thing on this has been things like caloric restriction. We did a study. We had rats living in an enriched environment where they had running wheels, they had rats of the opposite sex, et cetera, et cetera, et cetera. And the rats in the enriched environment got more neurons than the rats who lived in kind of a normal rat environment. And so we were really excited about this when we were trying to figure out what it was. Was it the sex? Was it the running wheel? Was it the enriched environment? Unfortunately, it turned out to be the running wheel as the most. We had all these great ideas about sexual activity, and maybe we get money from from the guys who are making Viagra, and but that didn't work. But that's a good question. I mean, the biggest thing here is aging, inflammation, and living in a low-rise environmental type environment. Sitting and watching television all day, every day, not getting much intellectual stimulation really impacts negatively on your stem cells. What I want to show you is that this system could be even more manipulated. So this is work from our own lab here in London. And if you go back here, one of the things that we showed many years ago was that in experimental animals who are subjected to stroke, what you see is this huge increase in the number of cells. So if I said that maybe you make 100, now maybe you make 10,000. Okay? In response to cerebrovascular damage. In the rat, if you look at it, so when you do a stroke in a rat, it's really pretty unpleasant. What you do is you basically ligate the middle cerebral artery you can either do it for 2 hours or you can do it permanently. It doesn't tend to make much of a difference. And what you end up with is, if you look at a rat brain, probably about 40% of one hemisphere is completely gone. Okay? Nevertheless, this stimulates a really potent neurogenic response. And you see millions of cells that migrate out from the subventricular zone and appear to try to go into the infarct. But the infarct's like a giant moon crater, right? And the cells, surprisingly, don't survive. There's no blood flow there. There's a giant hole in the head. And the cells do very badly. And if you look, it doesn't have much of a behavioral effect on the rats. They're quite impaired. Okay? And in 14 days, it's all over. The cells have stopped migrating. They've stopped being produced. It's now over. So we wondered if this was true in humans. And this was the study that really showed us something very interesting. So this was work that my lab did before I joined GE. So this woman was 84, and she had progressive vascular disease. And I think even if you're not a neuropathologist, you can see she's had all these small infarcts. And she had a big one here 2 weeks before she died. Okay, so we knew exactly what had happened. We couldn't give her bromodeoxyuridine. We didn't have approval at that time to do that. But she had donated her brain to us. This is if you're into human brains, it's one of the best brains we ever got. 4 hours after she was dead, we had her brain out and we were examining it. What we did is we used a series of different markers which only recognize stem cells. They don't recognize any other cells. And we looked within the region of damage and we compared it to tissue elsewhere in her brain where there was no cerebrovascular damage. Okay? Here you see her lateral ventricle. You see a couple of rare stem cells here on her healthy side. This is what's happened in her affected side. And to show you this in more detail, here you see the subventricular zone down here. And if you follow this series of photographs, there's just billions of cells migrating up into her region of damage. If you look over here, you don't see anything. If you look down here, you don't see anything. They are migrating specifically into a region of cerebrovascular damage. We then went on, so this was 2 weeks after death. We then went on and we looked at a whole series of brains out to 6 months after death, and we saw exactly the same thing. Unlike the rats where this happens over a 14-day period of time, this goes on for a long period of time in some patients. Now, what this means, we have no idea. You know, did these patients, because they had this really potent neurogenic response, did some of them functionally improve where they wouldn't have? Is this meaningless? Do these turn into new neurons, or are they all other support cells? Because we can't label these cells and because we can't do imaging, I have no idea. And nobody does. But biology is very parsimonious. You know, years ago we used to talk about how 90% of the genome was junk DNA. Remember that from biology? Junk DNA. It ain't junk. Why would your body in every cell, you know, resynthesize 3 billion base pairs of DNA every time a cell repairs if 90% of it is meaningless? You know, we'd be dead because that's just metabolically incredibly wasteful. Same thing for this. Why would our brains be making all of these cells if this was purely, you know, epiphenomenon, which I don't think it is? So unfortunately, when I joined GE, this work kind of went off to my colleagues and they didn't really pursue it very far. We at GE were trying to come up with a program together with 2 different pharma companies where we wanted to try to develop small molecule mimetics because we understand some of this. There's a series of vascular-derived cytokines, SDF1 and VEGF, which we think are really important for this process. And basically what we want to do is make small little drugs that people would take orally that would stimulate this process before they had a stroke. And unfortunately, that work never got funded. So it's still sitting there for some of you who'd like to work on this. Okay? All right. How am I doing on time, David?
Well, maybe 20 more minutes before we go into Q&A. It's all fascinating stuff.
You're joking.
But it's now
Oh, it's already been almost an hour. Okay.
Not quite
How about
an
30
hour.
minutes? All right. I'll speed
on
up.
you go.
Okay. So adult tissues in brain, in the bone marrow, in the intestine, in the hair, in skin, we've got some stem cell populations. But as I said, for a number of tissues and organs, we don't have anything. Okay? So where are we going to get cells that can make the tissues that we can't get from adult or fetal tissues? And the place is embryos, but even earlier embryos. Okay? So I told you that Jamie Thomson in 1998 managed to make cells the equivalent of the ones that Martin had made from mouse that gave rise to this revolution in mouse genetics. He now made these from human embryos. Actually, it wasn't him, it was an Israeli colleague named Joseph Ishkovitz. And in essence, what they did is they took embryos which had been created by in vitro fertilization. Louise Brown, who was born roughly 30 years ago this year, isn't it? Yeah. So IVF was a technology that was pioneered here in the UK by Bob Edwards and Patrick Steptoe. Bob won the Nobel Prize before he died, I think 2013, 12, 13, something like that, which was basically to take eggs from women, fertilize them with either partner or donor sperm, fertilize those in a dish, implant them on around day 3 into the woman's uterus, and with a success of roughly 20%, this gives rise to a viable pregnancy. Okay? So Louise in 1978 was the first quote-unquote test tube baby. Now so we started making embryos in the late 70s. By the mid-1980s, it was clear in this country that this raised a lot of ethical issues, because if you could now make embryos, maybe you could manipulate embryos, and maybe you could do genetic stuff on them, or maybe you could select for embryos. And you only have girls, we'll only have boys. Once genetics gets underway, maybe we'll only have embryos that have brown skin, or ones that have one blue eye and one green eye. So a very prominent Oxford philosopher named Mary Warnock was asked by the government to have a commission. She recruited a couple of biologists, in particular Anne McLaren, who was in Cambridge at that time, still was in Cambridge for many years. And they came up with a series of guidelines that basically would regulate how in vitro fertilization and all embryo research in the UK would be administered. Now, one of the first things that was obvious to people was that there were going to be a lot of embryos that were never going to be used clinically. When a woman is superovulated, given very powerful hormones to stimulate egg production, she doesn't have one egg stimulated, she may have 10, 20, or more, which are all removed. They're fertilized, and if a woman is relatively fecund and if she has access to good sperm, she may end up with 20 embryos at this point. It's not common, but it does happen. Let's say you end up with 10. 2 are implanted, the remaining high-quality embryos are frozen. So a couple may have 2, 5, 10, or more embryos frozen. If their pregnancy is unsuccessful, they'll usually have 2 more thawed and put back. If it's successful at some point, they stop. Or even if it's unsuccessful, many couples just stop. And what you end up with are embryos that are left over in the freezer, actually in liquid nitrogen. Now at King's, I worked with the IVF clinic there, we did 500-ish cycles a year. That's 500 rounds of this. We had rooms like this, full of tanks of liquid nitrogen full of embryos. Now, logistically, this is a nightmare. It's expensive because you constantly have to be topping up the tanks. And occasionally, somebody screws up and the wrong embryo goes back to the wrong couple. Okay? And sometimes this is pretty noticeable. So, it's, anything that deals with, how to make the system more efficient. So what the Warnock Commission, even back in the mid-'80s, decided was it should be permitted to allow couples to donate extra embryos, their embryos, to research if they wanted to. And so starting in 1990, we had the formation of the Human Fertilization and Embryology Authority, and they allowed for human embryos to be used for research, but only 5 research areas that were related to reproduction and fertility. Okay, and that's the way the life was from 1990. Jamie came along and made embryonic stem cell lines, as I'll show you in a moment. And we, because we'd been working on the mouse equivalents and we knew how to make mouse neurons, we knew how to make mouse islets, et cetera, we're tired of curing mice, we wanted to move to humans. So we tried to get access to the cells like everybody else in the world, and Jamie just basically said, bog off, you're one of 600 groups worldwide that want access to my cells. I don't have the resources to grow them for ourselves, let alone for everybody else in the world. So we, Austin Smith, Roger Peterson, and a whole bunch of us petitioned the British government to allow extension of the original 1990 Act to allow for 3 new research areas using human embryos, which would better understand human development, to create cell lines that could be used to treat human disease. So we were successful together with Austin. We were the first group in the world, actually, to get a license to derive human embryonic stem cells from donated human embryos. And actually, it's complicated but simple. This is what you look like when you are 6 days old. Okay? You're about 100 microns across. I like to point this out. There's no head, there's no arms, there's no legs. Okay? You're a 100 micron ball of cells, and you are predominantly what is called trophectoderm, which is the primordial placenta. So if you were in utero, you'd still be in the fallopian tubes. This is about 3 days prior to implantation. And this little population of cells down here is what turns into you and I. Okay? So this forms the fetus, this forms the placenta. If you microdissect this bit away from the rest of the blastocyst, as it's referred to, and you put it into culture, and we started out with what we had done with mice back in the 1990s, you end up with a colony of cells, which if you were successful in propagating this, so you cut this up into a whole bunch of little small clumps, you move it into some new cultures, and if you're lucky with an efficiency of about 10%, you can turn this into a cell line, a human embryonic stem cell line. Okay? It's called a cell line because it's basically from a single genome. Okay? So these guys are the same line, and it's different from this one. They look very much actually identical, but they have 2 completely different genomes. There'll be 2 completely different sexes, potentially. They could be 2 completely ethnic races, okay? Each cell line encapsulates the genome of that embryo. Okay? The two things that are really, really important about these cells is, one, if you can establish them, they will grow forever. They are immortal. Unlike cells that come from tissues, or even cells that come from really, really early 6-week or 4-week abortions, these cells will grow forever. And you can turn them into everything. Any cell type you want, because these cells, inherent within them, they have the capability of forming a complete and entire human being that goes through all the stages of development. From implantation in the uterine wall, formation of mesoderm, ectoderm, endoderm, making a liver, making a heart, making a brain, all the way through making 3 million blood cells every second. Okay? This is amazing. Yeah? And this is why Martin Martin did it first in the mouse. Jamie thought he should have gotten the Nobel Prize for this because he did it in the human. Most of us thought, well, he's just doing what was done in the mouse, and the mouse already got the Nobel Prize. So anyway. You're not Catholic by any chance, are you? I usually hear that when the Catholics are in the room. Let me give you some indication of what I mean by immortality. This is one of the first 3 cell lines that Jamie and Joseph made back in 1997. This cell line, H7, was one of the only cell lines that Bushbag used to allow American researchers to use in the US because it was derived before 2002. My cell lines here in the UK, which we derived in 2002 and 2003, American researchers would have gone to jail to use our cell lines. So this cell line was approved only because it was made before a certain date. Typical American foreign policy, right? This cell line's probably been used by more than 1,000 research groups worldwide. The number of cells that came from this one embryo that would have been destroyed anyway because it would have never been used clinically would fill this entire college. Okay? This cell line was made 20 years ago. At GE, we were growing 50 billion cells a week from this same cell line. And this cell line's been used by everybody in the world for 20 years. I know people have ethical concerns about using embryos in research, but if this doesn't answer at least part of that concern, you don't need to make 10,000 lines. Actually, we probably have made less than 5,000 worldwide over the last 15 years. These cell lines grow forever. And as I'll show you, you can still turn them into anything you want. Right. How do you take a cell that wants to turn into everything, and you only turn it into the cell type you want? How do you make a brain cell and not the other end of the spectrum? Right? It's bloody complicated. So Hannah, who was one of my PhD students many years ago, we were funded to try to make dopaminergic neurons, going back to the Parkinson's disease story. These are the stages we would have to go through in order to do this. Okay? You start with a stem cell in the brain. That's the earliest stem cell that arises in the brain, at 3-4 weeks after implantation. All the way through all the formations of cells that give you a specific dopaminergic neuron. Not a dopaminergic neuron that's generic, but a dopaminergic neuron that's anatomically specified that only dies in Parkinson's disease because not all dopa cells die. And if you don't have the right cell that comes from the right place, other dopa cells don't work. So this is called recapitulating ontogeny. Scientists always have to come up with something really like, wow, that sounds so cool. So my friend Lawrence Studer at Sloan Kettering in New York, who is doing this in human cells, it takes him 120 days to go from here to there, which is probably about how long it takes in your brain during fetal development to do exactly the same thing. Okay? If you screw up and you get here, but then you don't convert these cells into PDX3, this whole thing fails. And you have to go back and start over. So it's a step-by-step progression, and constantly you're going like this in terms of the number of cells you have and what their capability is. These cells can make pretty much any cell in the brain, at least in potential. These probably can make most DOPA cells. And then specification gets more and more and more concrete. Okay? When I joined GE, my boss said to me, I want you to make heart cells for drug discovery. Pharma every year makes compounds that basically go all the way through clinical development, being tested on animal cells, on tumor lines, in animals like hamsters and rabbits. You know, go through phase 1, phase 2, phase 3, they get licensed, they're going to be big blockbusters, and then lo and behold, people start to have adverse cardiac events. The most famous of which is a Merck drug called Vioxx. Vioxx killed 28,000 people by fatal arrhythmias. It was an antihistamine. It wasn't even like a really, really great drug, right? 28,000 people had fatal arrhythmias from a drug that if you had used human cells, you would have picked it up, and it wasn't picked up. So my boss said to me, because GE has a big program with pharma, let's make something that's better than what pharma is currently doing, and let's see if we can, if this works. So again, we took H7, the cell line I just showed you. We started out with huge numbers of undifferentiated cells, and in much the same way I just showed you, we converted them first into cardiac progenitors through a whole series of things and turned them into cardiac cells. This didn't take 120 days, it took 28, because the heart forms faster, and it's less specified. Does this look like heart? So imagine you have about 40 dishes that big. It's just a solid piece of contracting myocardium, each dish. Okay? It's about 80% heart cells, about 20% fibroblasts that just make nice goodies for the heart cells that keeps them happy and healthy. They beat in synchrony because you have ventricular cells, you have cells from the atrium, and you have nodal cells that pace it. They're all electrically coupled together, so you've got ion flow. So they're all contracting in synchrony. How many beats per minute? 70-ish. If you leave them out and they get cold, they slow down. It's kind of weird. If you look down the microscope, this looks like one solid piece of heart. This has never been in anything. It's never even been in an embryo, except for a 6-day blastocyst. Okay, so this has never been a heart. We made it from scratch. The amazing thing is you can take this, dissociate it down to single cells using enzymes, put it in a vial, stick it in liquid nitrogen, minus 185 centigrade, keep it there for 5 years, pull it out, put it back into a dish, and in 3 days it'll reform what I just showed you and it'll start contracting again. Okay? So pharma is slowly but surely beginning to use cells like this in their drug discovery. I have colleagues in Cambridge and the US are making liver cells, they're making kidney cells. These same cells, if you injected them into damaged heart, might survive if I put them onto a patch, synchronize them on the patch first, and then put it into the left ventricle of somebody who's had, let's say somebody who's in heart failure. They will synchronize to the existing ion current and beat rate of the donor, of the recipient heart, and at least in primates they repair. Chuck Murray in Seattle is probably 2 years from using almost identical cells like this in trials of patients in heart failure. Okay? So in all these, 15 years, the biggest problem for us has been this problem. You know, these cells want to make everything, and how do you only make them into the things you want? Okay, I have to move quickly now. I see David starting to get worried. So now there's labs all over the world and companies all over the world doing this, okay? I'm going to tell you 2 really quick stories to show you where this is going. I've showed you this bit, which is where we take a blastocyst, we isolate the inner cell mass, we make cells that can turn into everything, right? In 2002-3, when we started doing this, we could occasionally get embryos which would have genetic disorders. Okay? There's a program here called Pre-Implantation Genetic Diagnosis. These are couples who have a pre-existing genetic disorder in their family that they know about. So I have a friend, very good friend, who has Huntington's disease. Her father has Huntington's, If you have a parent with Huntington's, you have a 50% chance of getting that gene that will give you Huntington's. Unfortunately, Andrea and her brother Aaron both inherited dad's gene. They will both, roughly 61, 62, their brains will start to rot. Okay? We can screen embryos for Huntington's disease at the 8-cell stage, and we can identify embryos that have the Huntington mutation versus those that don't, and we only implant the ones who don't. The ones who are affected can be donated for research, and we can make a Huntington cell line. So in the UK, you can do this for Huntington's, you can do this for the BRCA mutations that cause breast cancer, we can do this for spinal muscular atrophy, we can do this for cystic fibrosis, and a few other diseases. Peter Brody, Susan Pickering, and I, we made the first disease-specific cell line in the world, which was from an embryo that had the most common mutation that causes cystic fibrosis, called the Delta 508 mutation. That was back in 2002. The problem is most diseases are not specifically what we would call single gene disorders, and the HFEA has been on top of it, has been very cautious about letting us do any kind of genetic identification of embryos if it's anything that's not a childhood disease. The BRCA mutations in Huntington's are the exception. So I wanted to make cell lines that had Alzheimer mutations or Parkinson's mutations, but I wasn't going to be able to get those from embryos. So how could I do that? So who else's anniversary is this year? Next to Louise Brown. Move ahead 20 years? Bah! Dolly. Dolly, baby. So Ian, Keith Campbell, Angela Ford, my friends up in Roslyn, made Dolly the sheep, right? And Dolly was made by taking a donor egg from a sheep, taking a cell from a 6-year-old Dolly, taking the nucleus out of the donor egg, putting Dolly's nucleus into the egg, making an embryo, implanting it into a sheep, and now you have Dolly 2, right? So Dolly existed previously. This was a cloned version of Dolly, okay? Ian's done a really great review this year. We now have cloned gerbils, we have cloned cows, we have cloned horses, dogs, pigs, rabbits, cats, and we even in the last month or two, we now have cloned primates. Okay, so we've cloned everything except us, which is illegal in most countries in the world. So that's called reproductive cloning, because you're actually making a living clone of a pre-existing person. There's another form of cloning called somatic cell nuclear transfer, or what was commonly called therapeutic cloning, where the idea is not to create another David, but to create a cell line that's genetically identical to David, right? So remember from the embryo, I said if we made a cell line from an embryo, the embryo would have all the genetic information that was in that embryo. If I can make a cell line from David, it will have all of his genetic information in there. And let's say, you know, he had an Alzheimer's mutation in his family that gave rise to a hereditary form of Alzheimer's disease. If I can use cloning to make a cell line from him, he will have that disease-causing gene in his cell line. And I can now make, neurons in a dish that have an Alzheimer's mutation in them, and I can try to figure out how does that mutation actually interfere with the normal functioning of the cells, how does it lead to Alzheimer's disease, how does it kill the cells, and can I prevent that from happening? So the whole idea was to use nuclear transfer or cloning to create disease-specific cell lines. Okay? Anybody have problems with that? Sounds good, right? One problem. I need eggs. Eggs. Give them over. My good friend in South Korea, Woo-Suk Wang, who was the master of this, he's still the only guy in the world who's successfully cloned dogs, tried to do this, used 2,000 fresh eggs from people in his lab, a number of very patriotic South Korean women who donated eggs to his research program, and he failed miserably. Okay, and ended up almost in jail because it was a huge scandal. So we got questioned about this because my colleague up in Newcastle, Lyle Armstrong, and I, at the same time, were putting in a license application in the HFEA to be able to do this. I wanted to make cell lines from individuals with known genetic disorders that I would not be able to get from embryos from IVF. And we were asked, well, if Woo-Suk Wang used 2,000 eggs and he's the master of the universe, how many eggs are you going to need? And I said, 10, 20,000. So how many women in this room would be willing to come in, be given powerful hormones, be super ovulated, and that means super ovulated, have a needle inserted through your uterus into your ovary, aspirate your eggs? And you don't get paid for this? I can't even give you taxi money. Anybody? Yeah, of course not. So we said to the government, well, we don't want to use human eggs. And they were like, so what are you talking about? We said, well, let's use cow eggs. Let's use sheep eggs. Let's use any eggs. And we got a lot of flack for this. It took us 4 years. But, we got support from people like him. Gordo was pretty happy about this because he had a child with cystic fibrosis, right? So here's the whole idea, right? You take eggs from cows. The cows are already being killed, so no additional cows are being harmed in this process, right? We're getting eggs from the slaughterhouse, from the abattoir. You remove the cow nucleus. Our argument is what makes a cow is this. Once the cow's chromosomes aren't there anymore, it's not a cow. It's a nothing. It has cow mitochondria. I will claim that. But it ain't a cow. And if I take a cell from Gordo, a whole human cell, not just a nucleus, put into this what used to be cow egg, which is now a nothing egg, and I make an embryo, this has to be a human embryo. It's not a cowboy. Okay? It is a human embryo, and the cell line that's derived from that is human, with cow mitochondria, which over time will be replaced by human mitochondria. Caroline Flint said this is the most monstrous thing I've ever heard of. The British government's got to ban this, blah, 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 blah. Parliament, and so we had for 2 years we had a huge row. We went through a consultation in Parliament, went through a consultation with the HFEA. Everybody finally agreed that these were human embryos, even if they were derived from what used to be a cow egg or a pig egg or a sheep egg. And then the government decided, well, this is still kind of weird, so we need a new bill. So then we spent 2 more years coming up with a bill called the new Embryo Bill of 2008. So I can say with some pride that we actually took a bill to Parliament that is the existing embryo legislation in this country.
Where did the human mitochondria come from?
Come from the human cell, because I didn't put in a human nucleus, I put in a whole cell.
Right, okay.
And so mitochondria are made up of genes that come from the nucleus and genes that come from the mitochondria. So if there's no cow nucleus, over time, originally the mitochondria will be hybrid, but ultimately they get weeded out. And ultimately, I don't know how many passages it would take. We never actually did this, and I'll tell you why in a second. But you can see on the day that the bill was debated and ultimately passed in Parliament, there were quite a few people who didn't like this. My favorite one was The Sun. When we said we wanted to do this, The Sun had a headline with a guy in a business suit, very well dressed, with a cow head and a briefcase, and it said "mutants." It's one of the few newspaper headlines I really like. Anyway, this went on. This was 4 years of my life. The reason for bringing this up is actually not at about the same time we were doing this. There was a really, really bright guy in Japan who said, there's got to be a better way to do this, right? So Sheena figured out that there has to be something unique about these cells that allow them to be immortal and that they can turn into everything. And so he began to look at what's genetically different about these cells versus any of the other cells that they turn into. And obviously it's genetic, right? At least at some level. So initially I think he identified something like 50 genes that were really highly overexpressed in these cells versus these guys. And again, starting in the mouse, because it's always easier to do and faster to do things in mice, he took these 50 genes and he put them into a fibroblast and he made something that looked like a pluripotent cell with an efficiency of about 1 in 5 million, okay, which is ridiculously low. So then the question was, well, do I need all 50? So let's try 49, let's try 46, let's try 43. 4 genes. With 4 genes, I can take every one of you in this room and I can make an equivalent cell line to you as an embryo. Every one of you in 6 months.
What do you mean by 4 genes?
4 genes. I put, using vectors, using messenger RNA, using protein, I take OCT4, SOX2, NANOG, and a gene that varies depending on the group. I put into a fibroblast, and with an efficiency of about 1 in 1,000, I can make 8 or 10 or 100 clones that will turn into these cells, which are all you. They're your cells. It's the equivalent of the cells I would take from an embryo.
Except for the methylation patterns don't change completely, do they?
That's very controversial. So the point that this gentleman's making is that these cells are derived from an embryo. These cells come either from a fibroblast or from a blood cell, a muscle cell, a skin cell. They may have genes that are still open from the source of the cell that they come from, where these cells won't. So whether they're truly fully equivalent to embryonic stem cells is still a matter of contention. I'm trying to be as nice as I can, because I think that there's still some differences about these cells that are different than these. Most of the field would argue that the differences are slight. Certainly for working in a dish, it's probably fine. If you want to use these cells for therapy, that may be a little more problematic. But in principle, they look like, they smell like, they quack like pluripotent cells. Okay? Now think about this. If you're, instead of waiting for a bone marrow transplant or an organ transplant from somebody else, I could make a cell line from you, convert it into the cells I just showed you, and you get those back without immunosuppression and without rejection. It'll probably cost $1 million or $2 million per patient to do that, but it's at least feasible. Okay? Even now, we have technology that we can take a somatic cell, and instead of going through this phase, we can take fibroblasts and we can convert them directly into neurons or directly into heart cells without going through this IPS phase. This completely turns developmental biology on its head. The idea, that everything's going down this track in one direction, and development proceeds and you can never come back. I mean, this won the Nobel Prize in 2013, and it's not surprising it did. And this has opened the door to American researchers because there's no longer any embryo. Okay? So in 5 years, you will each have your own cell line sitting at your refrigerator at home. Okay. All right, I really got to move now. All right, so this is a short shopping list of diseases for which we think cell replacement doesn't have to be necessarily stem cells, but cell replacement therapy will have applications. And many of these, like diabetes, myocardial infarction, blindness, baldness we could talk about, are already in the clinic. Wound healing, we do a lot of stuff with replacement skin. Many of you will have seen a child a few weeks ago who had epidermal lysis bullosa. Again, that was partially research that we did at King's together with the group in Italy. Spinal cord injury, again, we're in phase 2 trials now. This is all ramping ahead very quickly. And again, the number of cells that we can, different cell populations, there's no such thing as a perfect cell population. It's what do I need? How many cells do I need? What stage of development do I need them? Do I have to grow enough cells to treat 1,000 patients at once? Can I take a small population of cells, grow enough of them to treat that patient and that patient alone? We're still embryonic in this sense of manufacturing cells for cell therapy. But as I said, go into clinic. It's called clinicaltrials.gov, and go in and have a look. I mean, we're cells for Crohn's disease, inflammatory bowel disease, alopecia, epidermal lysis bullosa, multiple sclerosis, muscular dystrophy, everything you can think of. We're in phase 1-2 trials now, and some things are getting approved in the clinic. So this just basically says what I just said. On the one hand, we might want to grow giant, 10,000 liter bioreactors where we're growing enough cells to treat everybody who needs, cartilage repair. On the other hand, what's happening with this new generation of cancer treatment, this is called autologous cell therapy, and we're treating individual patients with their own cells. It's still very labor intensive, it's still incredibly expensive, and we still have no idea what we're doing. We're really pissing in the dark, as my grandmother would say. She did talk like that, so don't be surprised. But in essence, it's quite a simple procedure, but we don't really even understand how much of this procedure we need to do and how much we might be able to shorten or alleviate. But in principle, what happens is in cancer, for some reason, with the exception of melanoma, the immune system in some way is completely silenced. Tumors, particularly solid tumors, should be recognized by the immune system because your immune system, your MHCs, are displaying tumor antigens, which in a normal setting would be recognized as being foreign. But they're not. So the immune system is suppressed. Somehow we have to figure out how to reactivate this. So starting back in the 80s, Steve Rosenberg and a guy from Israel, Ellerig, who did his postdoc with Steve, began to think about, well, how do we unleash the immune system? How do we get rid of this suppression? And they came up with this idea. Basically, you harvest white blood cells. So you sit in a machine. You have one set of lines that pull blood out of you. It goes through a processing machine, and you get everything except your white blood cells back. It takes about 3 hours. It's called apheresis. We then use separation technology to pull out very specific subsets of white blood cells. So we get rid of the T regulatory cells, the T helper cells, and we only collect CD4, CD8 positive cells. Now these cells, because they come from a patient with cancer, these cells are just sitting there dead in the water. They're not doing anything. So the first thing we have to do is activate them. We have to convince them they want to go after tumors. So you have to drive them to begin to proliferate and to divide. We then generally engineer them in some way so that they're now cancer specific. And this is really difficult. How do you find surface receptors on cancer cells that are unique and not expressed on normal tissues? Because as I'll show you in a minute, these cells, when we put them back, they are lethal. I mean, these things will kill you in less than a day. You never thought your immune system could do this. We grow them up, and usually we grow as many cells as we can, and then we put them all back. Okay? When Steve and Ellerig started first talking about this, everybody thought they were crazy. And most of the patients, they either saw only a partial response, some of the tumors died, most of them didn't, or occasionally, like 1 in 20 patients, they would get a complete and curative response. But they didn't understand why. But other people piled in on this, in particular Carl June and Bruce Levine at Penn, and Michel Sandelin and his team at Sloan Kettering. With Carl, Carl originally was working on HIV. His wife died of breast cancer at very young, and he decided he was going to devote his life to cancer therapy. So Carl and Michel started probably back in the 1980s, early 90s, to try to figure out how to engineer white blood cells to attack cancer. Starting in about 2011-12, this hit the fan like there's no tomorrow. Bruce and his group, so there's a disease called B-cell lymphoma. You have T cells and you have B cells, which are two arms of your immune system. B cells, when they become cancerous, will grow like crazy. And there is no therapy short of bone marrow transplantation that's curative. You can go on really powerful cancer drugs. You can kill most of the tumor, but it will always come back. Most patients don't survive more than 2 to 3 years. Okay? So in B-cell lymphoma, it's pretty true in leukemias as well. Bruce and Carl made what's called CAR T cells. CARs stand for chimeric antigen receptor. These are artificial receptors for a protein called CD19 that's only expressed on B cells. They engineered these cells, they grew them up in our bioreactor, our GE bioreactor. That's how I got to know these guys. Put the cells back into the patients, and in this paper that was published in 2011, 5 out of 5 patients who got their own CAR T cells back went into complete and total remission. Total remission. At the level of the most sensitive test that you can look for in blood, they had no B cells left whatsoever, tumor or otherwise. Emma is the poster child of this. She was dead. She was 5 years old. I saw her at the time she got infused, having spent 3 years in an intensive care unit around bone marrow and solid organ transplantation before I did my PhD. This child wouldn't have lived another week. Totally emaciated, no hair, no teeth, jaundice, dead. In 2 days, she was cured. She's now the most obnoxious 12-year-old you've ever seen. Her parents hate her. She's total pain. Michelle's data is even more impressive. 12 out of 14, complete remission. in the same disease, same target, same everything. Now, we think about 80%, now we've done about 300, maybe 400 patients now since 2012. About 80% of patients have what we call a complete response. That is, they have no disease at 6 months. We think about 30% of those people will revert because they have cells that were B-cell tumors which did not express CD19. We kill off 99.99% of the tumor, a few cells evade, and they come back. We now have to go after them with another CAR that's not against CD19. So now we're starting to do second, third, fourth, fifth generation CARs looking for other antigens. The problem in all of this is that these cells are lethal. Almost every patient that we give back their own cells to, we have to put them back on life support. We have to put them back in the intensive care unit because, and the more tumor you have, the more dramatic a response you get. We have killed patients doing this. Now these people, these are all end-stage patients. They would all be dead otherwise, okay? But we have killed people in the process of giving them back their own cells. But about 50% of people in these early trials we think have a curative response. That is, their tumors should never come back. The big challenge now is we move towards solid tumors. Can we do this in prostate and breast and colorectal cancer? Can we do this in brain tumors? We just don't know. There's issues around what proteins do you go after? How do I know that a protein that's only expressed on a brain cell in a glioma is not expressed by the rest of the brain? If it is, I'm going to fry the whole brain. These cells make really potent things like interferon and tumor necrosis factor. That's why they're so deadly. So target identification, target selectivity. We may have to have CARs that recognize 4 different targets, and the cells only activate if all 4 of those are engaged. This is going to get really complex. Okay? But this is the hottest thing in cancer since I don't know when. Science called it the breakthrough of 2013. Novartis put money into this. My friends at Sloan Kettering, Celgene just acquired their company. Now think about this. My friend Michel and his partner Isabel. Isabel runs their cell therapy manufacturing facility. They're 2 of 4 founders of Juno. Juno just got acquired by Celgene for $9 billion. How much do the 2 of them get from this? I can't even imagine how much money that must be. Another friend of mine works for a competing company called Kite. He was scientist number 4. They got acquired by Gilead for $12 billion. I would love to see what his options look like. Jesus. Anyway, that's cancer. If we can do this in solid tumors, this is just going to be explosive. Whether we can afford to do it is a different issue. These therapies are roughly $500,000 per patient currently. Okay? All right. I'm going to give you 5 minutes of where we're at kind of now, but these are really new emerging things. Then I'm going to talk 5 minutes more about things that are way out there. Okay? So what I do as a consultant for a lot of the companies I work for is I do what's called horizon scanning. I do blue sky scanning. You know, what's out there that people should think about investing in? And when investors come to me and say, you know, I've seen this wacko company, what do you think about them? These are the kind of things I think about. So, and not all of them I find that interesting. This whole thing about omics is becoming ridiculous, okay? So this gentleman talked about methylation states. So you probably within a couple of years in your purse, in your handbag, your wallet, you'll carry around your DNA sequence on a little chip. That when you go see your doctor, he'll slide it in and it'll pull up 1,000 susceptibility genes that you have for a whole variety of diseases, some other things that are important, but basically your whole genome will be there, okay? Right now it costs about $1,000 and a day to sequence 98% of your genome. Next year it'll be about $100 in half a day. That's just your genome. That doesn't tell you anything, that just tells you what you've inherited. What about which genes are expressed in different tissues and at different times in your life? So now you have basically mRNA data, expression data. That only tells you which genes are on in certain tissues and at certain times of your life. It doesn't tell you actually about the proteins that are made, because even from one gene you make different proteins, something we call alternative splicing. So from the same gene, you know, there may be 10 or 15 or 100 different varieties of that protein. We don't know that from reading your sequence. We only know it if we look for all the proteins that you make. So that's called your proteome. Then there's your metabolome. We all metabolize stuff differently. We have different enzymes in our body that work at different rates. Prototypical example is there's certain Asians, unfortunately, that if you give them half a beer, they turn beet red and fall over. Sometimes take their clothes off as well, but that's, you know, so they metabolize ethanol very differently than Caucasians. You know, I have friends who have gluten allergies, you know, and so they metabolize gluten very, so it just goes on and on and on. You have bacteria in your intestine in particular that everybody metabolizes food differently, not because of genes, but because of the bugs in your body. Okay? Half your health and your diet is due to the bugs that are inside you, not to your genome. So this is becoming, on an individual basis, just a ridiculous amount of information that we don't have the capability to analyze now. I mean, if you go to 23andMe, you'll get a readout, I think, on how many have done 23andMe? How many genes do you get?
I can't remember.
It's like 100 or something, right? It's not many. Okay? And who owns that data? Do you?
Everything on file.
Yeah. What else have they done with it?
I guess a little poll.
Well, they sold 1 million genomes to Roche. So I don't know if your genomes were part of that or not. But, you know, with 23andMe, that's almost the kind of the limit at what we can look at. Now we're talking about having access to your whole genome, your epigenome, your metabolome, blah, 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 epigenome, et cetera, et cetera. We don't have the computing power to do this for one person, let alone for doing it if you want to look across 1 million patients. That's where people like Deepmind come in. Okay? How many of you know about Deepmind? Everybody? You know about AlphaGo Zero, right? AlphaGo Zero. I mean, if a machine can learn how to play Go in 2 days and beat the world champion 100 to 1, 100 to 0, this is the kind of computing power you need to put to this. Now, surprisingly, I was up at Hingston at the Genome Center earlier last year, and I sat through a whole day of cancer genomics. And if you ask them, okay, so these guys are looking at breast, these guys are looking at prostate, these guys are looking at breast, these guys are looking colorectal. We have 100 patient samples. We've sequenced to entirety their tumor versus their non-tumor tissues, and you know, we have our own proprietary database, and you can get access to it. Do you talk to these other guys over here? No. Why not? Well, they're doing their thing, and we're doing ours. So I sat through 12 different talks. Everybody's doing exactly the same thing. Nobody's talking to each other. And it was all genomics. There was nothing else. Okay? I'll come back to this in a moment because it's really important. This has got to change because the amount of data that we're starting to generate on a per patient basis is just astronomical. And yet when we start to do large, huge, you know, there's the 100,000 genome project here in the UK. Everybody wants to get 100, 200, 500,000 patients into their samples. And if we don't have huge amounts of machine learning, this data is just going to be indecipherable. Okay? And very few people, except people like Deepmind, even IBM Watson, are not really talking about. Deepmind's probably the leader in this. Wearables. How many wear wearables? So I get asked almost every day, how many steps do you take? I don't wear anything. I walk 6 miles, right? Well, how many steps is that? I don't know. But if you don't know how many steps, how do you know how many calories you're burning? I think I can tell, you know? I mean, you know, so everything's wearables, right? So how much you sleep, how many times you snore, you know, I don't know. What do people do with all this information? I haven't quite figured this all out. Okay, the only thing I've heard recently that made any sense to me about wearables, but this technology is coming, okay? And people want it, is a guy who's a psychiatrist who has a really, really severe patient population that's got extreme bipolar disease. These are patients who have extreme mania and then almost catatonic depression. And he'll see them every 6 or 8 months, but he has no idea what happens to them in the intervening time. He's put them all on a Fitbit, is that right? And every week he pulls up their activity logs, and he's got it, so he's on one screen he pulls up 100 patients at a shot. And he just looks across and looks for the outliers. He finds patients that just don't move for a week versus those that don't sit still for a week. And he knows those are the guys he needs to bring in. This is one of the few examples that I've heard of that really makes sense to me about this technology. I'm sure you guys have got a multitude of uses for it. I kind of stay away from it. I'm not sure I want to know that much about myself. This is becoming a really hot topic. Have you heard of CRISPR? I'm CRISPR'd out. I don't know about you guys. I've had enough of CRISPR. CRISPR is just a technique. It's a gene editing technique. We've been doing gene editing for 30 years. I mean, we gave a prize, a Nobel Prize for homologous recombination to work that was done in the early 80s, which was gene editing, okay? The advantage of CRISPR, like iPS cells, is that CRISPR democratizes gene editing. It's simple, it's cheap, it's easy. Everybody, including your poodle, can do gene editing now, okay? And it's headed toward the clinic. And so we're going to have a lot of gene driver technology, which is basically the ability to manipulate organisms where they're not going to be considered GM. Already in America, you can get apples and mushrooms that don't turn brown. I mean, that's really important. You know, more importantly, we have Zika. We have mosquitoes that can't transmit Zika because they're basically, when they mate with a female who has Zika, they're sterile. They have no offspring. So you're going to start to see what a lot of what's called gene driver technology, which is basically engineering organisms for some proposed wonderful thing. My big one on this, you guys can, I've already put in a patent for this, so don't worry about it too much. But I want to make designer cats. Okay, so I want to make fluorescent cats where they have like a blue head, a green body, red legs, and yellow paws, and they glow in the dark. All right? I mean, that's got to be as good as, you know, mushrooms that don't turn brown, right? I mean, this is what you're going to see a lot of, is a lot of driver technology where we're going to be able to create, using CRISPR, organisms that overexpress different genes in different ways that we really haven't thought of. And the fluorescent idea isn't so crazy if you were to hook that up to a gene, a real significant gene of interest, and make something that gives you a reporter system. This goes hand in hand with genetic engineering of embryos, though. And this is a really big debate, both here in the UK, but more importantly in the US, because in the US, they don't have the kind of guidelines we have around embryo research here. And so in the US, there's a lot of consternation about this, despite the fact that in at least 25 states in the US, there's no law preventing me from cloning you. I could clone you, your sister, your brother, all of you. It's not illegal. Here, I'd go to prison for 10 years, okay? So the fact that we're even having this debate about genetic engineering of embryos, when in essence in America you can do anything you want with an embryo, is actually quite surprising, because individual states can mandate this separate from the federal government. But this technology, I think CRISPR is just the tip of the iceberg. I think we're going to see stuff that is, you know, in 5 years, CRISPR is going to be like zinc fingers or talons were a couple of years ago. Pharmacological enhancement. Any of you take cognitive enhancing drugs? Are they good? Do they work?
Yeah.
Yeah?
But you need to know how to do it.
What do you take? What is it? Metrophenate? No.
No.
Modafinil?
No.
Yeah. No? Okay.
I won't even consider it now.
Okay. So I have friends who take it and they claim it helps. I tried it and I just couldn't feel any effects. I mean, cocaine's a cognitive enhancing drug, right? For a while. You know, this is going to be a big deal. I mean, already, my friend John Harris, who's a philosopher in Manchester, talks about, well, if your kid's competing for a place at Oxford and the kid next to him is taking cognitive enhancers and your kid's not, is he being disadvantaged? And, you know, should everybody be able to have access to statins? Should we just make them over the counter? Et cetera, et cetera. So, you know, pharmacological enhancement, I think, will become a bigger deal. Probably more important in my consideration is technological enhancement. You know, we have glasses and we have hearing aids and other things. I think soon we're going to have more than that. Okay. Robots. Who likes robots? Did you see the dog from Boston Robotics that can open the door? That was cool. I like that. I think those dogs are really cool. I can't imagine why Google sold them. I think that's one of the coolest companies. Anyway, robots in healthcare. You know, nurses, surgical assistants. My friends talk about in the future, you know, surgery, there'll be nobody in the room except the robots, and the physicians will all be outside watching and you know, kind of helping the robots, but basically the robots will just do everything. And I'll show you one example of that in a minute. But I think, you know, even in Japan, where you've got a drastically aging population, having robotic assistants in hospitals, in medical settings, I think is going to be huge. Synthetic organisms, this is another one that's very controversial. People like Ventnor and synthetic genomics wanting to make species of organisms that never existed, making new designer genes, designer amino acids, designer proteins that never existed. I met a guy who's a prof in Ljubljana, and he basically has developed proteins to the extent where he can, by putting in different amino acids that don't naturally exist, he can basically fold proteins into any shape he wants. So tetrahedrons, squares, polymer, all sorts of cool stuff, just by manipulating the code. So I think you're going to see a lot of novel protein design that are going to take things, for example, like, let's say insulin, which has to be stored at a certain temperature and a certain acidity. Play around with it, you don't actually change the structure of insulin and its therapy, but you change the physical properties of the protein itself. There's a big group at MIT that's doing novel protein design, probably the best in the world. And there's a big, big thing on this. Synthetic organisms, it's the common one. We want to make bacteria that eat oil. There's a huge oil spill in China last 2 weeks ago. That would be perfect. This one is big in the US again, not so much here. The US has really dropped the bag on allowing what many of us in the field would consider to be unethical and dubious cell and stem cell treatments to basically go ahead with very little oversight. Trump is pushing very hard on this right to passage bill, which would basically allow patients access to any therapy that they can find, whether it's been proven or not, whether it's been through clinical trial, as long as they're willing to take onus for any safety concerns. We've already seen this go badly wrong in Florida. People were injecting adipose-derived tissue into patients with blindness, and they basically got fat that continued to grow in their eyes and basically got blind from this. A whole bunch of people have been treated with stuff in India, in Russia, that continue to grow after implantation. I think the last thing any of us want to see is a relaxation of guidelines that say if it hasn't been through a phase 1, phase 2 trial minimally. I mean, the oncology stuff got expedited approval. Novartis got approval on a phase 2, and Bluebird got approval on a phase 2. But the efficacy there is so dramatic. We know what the safety risks are. And the patients are getting back their own cells. So we're not so concerned about something around the cells. It's more about the clinical efficacy of the cells and how dangerous they are to work with. This is one of my favorite ones. Clinical use of artificial gametes. Okay. So one quick funny story. So when the IPS stuff came along, and Yamanaka's work showed that in humans, I could take any of you and I can make a pluripotent cell line from you, I got a call from Bush's head of the Council for Bioethics in the US, a guy from Stanford named William Hurlburt, who I've had a really fun relationship with. He's adamantly opposed to everything I want to do, but we actually get on quite well. So William called me out and he said, "Stephen, this is fantastic. George is so happy, For once we have stem cell stuff where there aren't any ethical considerations at all." And I said, "Really? None at all?" He said, "Yeah." And I said, "Really, William, you think so?" And he said, "Yes, because there's no embryos." I said, "Okay. Well, if these cells can make every cell type in the body, you're all right with that?" He said, "Yeah, of course. What's the problem?" And I said, "Well, if I'm a guy, right?" He said, "You are." And I said, "So I'm XY, right?" And he goes, "Yeah." I said, "So if I'm XY, that means I can, from my cells, I can make sperm." And he says, "Well, you already make sperm." I said, yeah, just wait a second. "So I can make sperm from these cells, right?" He says, "Yes." I said, "But because I'm also X, I can also make eggs." And he's like, "What?" I said, "Yes. Because I'm XY, I can make eggs and I can make sperm from my cell line. And then I can fertilize my own eggs with my own sperm." What would that be, William?
Mini-me?
Twin, brother, sister, I don't know. What is it, biologically? And he's like, "Oh, shit, we didn't think of that." I said, "Even better. I'm sure George is really in favor of this." He said, "What's next?" I said, "If me and my boyfriend want to have a baby together, we now can." So gay men could biologically conceive babies together. They would just need to rent a womb, right? He's like, "Oh man, what are you talking about?" I said, "William, this was your idea, not mine." Scenario number 3 is you're in a restaurant, and as in my local, about 2 weeks ago, I was sitting there on a Sunday evening, and one of the women at the pub came over, and she said, "Do you know who you're sitting next to?" And I said, "No, I'm working. Why?" And it was Hugh Grant. And I was like, "What? Who's Hugh Grant?" So she finally explained it to me. But anyway, he got up and went to the toilet at one point. And what I thought of was he was sitting there, and his beer glass was sitting there, and I thought, So I could have gone over, taken his buccal cells from the top of his glass, made his eggs and sperm, and sold them on eBay. Now, could he stop me from doing that? Do we have any lawyers in the room? Is that legal? Is that identity theft? Number 4, I was invited to a conference in Turin to the Italian IVF community. And I was very nervous because every time I go to Italy, I have things thrown at me and people shouting at me. So I said to my friend, what should I talk about? He said, "I want you to talk about everything." So I talked about the cloning stuff because this is when we were trying to get this bill through Parliament. And all these Italian physicians turned on me and said, "Yeah, we don't need this cloning crap. We can do IVF, or we can do IPS, blah, blah, blah. We can make, cells from everybody." And I said, "That's a very good point. You can make cells from everyone." And they said, "Yeah, so what's the problem in that?" And I said, "Think of it. Not only can I make cells from all of you guys, in principle, I can make cells from anybody who's ever existed. All I need is DNA." They're all sitting there looking. I said, "Think of all the famous people who have ever been to Turin." And I'm looking over at my friend, the professor, who was the first guy to do IVF in Italy, and he's going, "No." So I said, "Napoleon." I didn't talk about the shroud. Because with the shroud, you could make the eggs and sperm of Jesus, if that was Jesus, right? So this is going to become a big deal. In the UK at the moment, you can use gametes derived from stem cells. If you have a license and you can make embryos, you cannot implant them. Okay? But it's legal here to do the work. If you can prove to the I don't know if anybody has a license. I don't think so. But you could, in principle, do the work here. I know because I helped draft the bill. Okay, let's talk about 3 more things. So this is, how are we doing on time, David?
Well, that's all fascinating. If you're quick, we'll let you go.
I'll be quick. So these, when I was leaving GE, these were the things that we were looking at. I'll talk about bioprinting for a second. I want to talk about this one here, because this is, I think, an area of science that, for whatever reason, scientists have just been really stupid about. We have just ignored this. What's the microbiome? The microbiome are the trillions and trillions and trillions of bugs, viruses, fungi that live in us, on us, throughout us. Okay? We've always thought that these things are just bad for us, right? When you get an ulcer, it's because you've got, H. pylori in your stomach. If you have inflammatory bowel disease, because you've got bad bugs in your gut. When you go to third world countries and you get diarrhea, it's because you've gotten bacterial infections, and we just destroy ourselves with antibiotics every day. In the last 5 years, this has become, again, one of the hottest areas in science. We are more of them than we are us. You have 10 times more bugs in you than you have cells. We're just a giant Petri dish. Okay? And particularly in the gut, we have no idea what's in our gut because we can't grow any of this stuff. These are anaerobes. They need they live in an oxygen-free environment. We have no idea what they live on. We have no idea what kind they are, what they need to survive. So up until the advent of high throughput genome sequencing, we had no tools to try to understand this. Now we can take a stool sample from anybody, subject it to NGS, and I can tell you in a day how many thousands of different bugs live inside you. I can't tell you what they do, but I can tell you what they are. Okay? In people, this has been really important in people who have Clostridium diff, which is a really nasty bacterial infection, which when antibiotics fail, you are doomed. There's a couple of antibiotics that work, but people who have really bad C. diff infections, they're basically they have no antibiotics that work anymore. Okay, so they have no treatment. The standard treatment now is to take a fecal sample from generally a related sibling, if possible, but if necessary, from a healthy donor, and you get a poop transplant. okay? Generally, you go down the top, past the stomach with an NG tube, and you just take a slurry of poop and you inject it in. 98% of people resolve without antibiotics, and it never comes back from a shit transplant. Okay. We've genetically engineered mice to be really, really lean, so they overexpress leptin, okay? These mice never get fat. You can rear them on McDonald's and they never get fat. They look like greyhound mice. There's another set of mice who get really, really fat called OB mice, and no matter what you feed them, they get fat as hell. If you switch their microbiomes, they convert to the other species. The fat mice get lean, the lean mice get fat. I read today where a woman who got a poop transplant from her sister became grossly obese because her sister was obese. So these bugs are going to be more important in disease prevention and disease progression than we ever thought. If you take mice who are engineered to develop Parkinson's disease because they overexpress a Parkinson's disease a mutant gene, and you take them before they will, so you got two sets of mice, and let's say at 10 months the mice begin to be Parkinsonian. If you take these mice and give them a stool sample from a patient with Parkinson's disease at 6 months, they convert to Parkinson's disease faster than these guys do. So there's something in the microbiome of Parkinson's patients that, at least in mice, cause them to develop Parkinson's disease faster. Whether you respond to a chemotherapeutic agent has always been thought as to whether or not you express the particular mutation that drug works for. There are patients, when you compare them, that respond and don't respond, yet they have the same expression pattern. It appears that their microbiome decides whether or not they metabolize the drug appropriately or not. So we are a giant petri dish, and we've got to start to pay more attention to this. And this is going to be the next big thing, I think, in healthcare, is understanding this in your stomach, in your ears, in your nose, in your mouth, et cetera, et cetera, et cetera. And we've just ignored this for years. It's been really stupid. And these are just, anyway, let me move on before David really starts yelling at me. 3D bioprinting. This is basically around making tissue constructs. And it fits very well within what we've been doing historically, So replacement therapy is things like erythropoietin, insulin. Then we made monoclonal antibodies, so now we have Herceptin and a whole bunch of Yervoy and a whole bunch of different monoclonals. I've told you a lot about cell-based therapies. Now we want to move toward fabricating tissues and tissue-like organs. The reason for that is painfully obvious. This is the number of patients, this slide's a little outdated, number of patients who are waiting organ transplantation, and the number of donors. And remember, you can get a kidney from a living donor. You can also potentially get at least part of a liver from a living donor. The vast majority of these rely on cadaveric tissues, particularly islets and heart. So something like 80% of people waiting transplant, even for bone marrow transplants, never get transplanted. So can we make replacement tissues? So this is work, I think, from Tony Atali's group in Wake Forest. It's actually really quite simple. You have basically a 3D printer, and instead of inks, you have cells. And you have a computer program that basically takes the structure of what you need, converts it into a printing program, and then you just print it. This is pretty simple. I mean, this is basically spray painting cells onto a collagen scaffold. What we wanted to do was make a left ventricular patch, okay, that would mimic what you would see in the heart. So here what you do, you take a tissue section, say this is a piece of left ventricle that's maybe a centimeter thick, and you slice it into very, very thin 10 micron sections, and then using high content imaging, you stain for all the different cell types, and you basically create a map of this is a cardiomyocyte, this is an endothelial cell, this is a smooth muscle cell, there's another smooth muscle cell, here's another cardiomyocyte, et cetera, et cetera. So you build up a three-dimensional map, but it's topologically precise. You then make a CAD diagram. This has actually been done. This is printing this with plastic, but this could be cells. Okay? Then you need a bioreactor to grow it up in. Have to figure out how we're going to vascularize these things in the bioreactor, but that's really the hardest part. Making this stuff, we have all the technology to do this. Okay? So people are already making a lot of replacement tissues, but they're basically structural ones like noses, larynxes, ears, a bit of bone. This one I really like, being a neurobiologist. This is based on technology we pioneered back in Minnesota in the 80s, and it's called electrocorticography. This is basically brain mapping, okay? It's been used predominantly in patients who have seizures, where you want to precisely map out where their seizures are coming from and compare that to other surrounding high real estate areas. This is generally done in patients who have left temporal lobe epilepsy. Almost everybody in this room will be left temporal lobe dominant. That is, in your left temporal lobe is your speech center, your ability to navigate in space three-dimensionally, control motor movement, speech recognition. Most of your higher faculty cognitive processing all occurs in the left temporal lobe. This is a young smart chap named Eddie Chang. He's a neurosurgeon at UCSF. And so Eddie has really taken what we were doing in Minnesota and taken this to the next level. We were working with about 80 electrodes. He has more than 1,000 electrode grids now that he sews onto the surface of the brain of the patient. And over a period of about 2 weeks, you take them off anticonvulsants, you record their seizures, but at the same time, every second of every day, you're monitoring them and you're testing them. You're reading to them, having them read, you're showing them sounds. You have a room full of psychologists and people testing these patients. And basically what you're doing is you're building up a map of all of the functioning under the electrodes. You can't test away from the electrodes, but under the electrodes you can map any of this stuff. Okay? What Eddie has shown, and this is a paper in Science, is that if you look at speech recognition, there's a series of neurons that respond specifically to really hard sounds like da, pa, ka, ra, right? But they don't respond, or generally don't respond, to any of the other sounds. You have another set of guys here, which are completely different place in the brain, that don't really respond to the really hard sounds, but respond preferentially to oo, I, e, ah. Okay? And then you have some guys over here that are pretty random. But basically what he's doing is he's building up at about the 1,000 neuron level a map of how we process speech. Okay? What he would like to do is understand what the electrical activity is. So if you understand this, what the amplitude, wavelength, electrical stuff, right? Whatever, action potential stuff. Frequency, amplitude, et cetera, if you had a stimulator and you stimulated this part of the brain, would you hear sound in the absence of sound? Not through your ears, but in your brain. You should, right? Because the wetware is there. It's like people who are retinally blind but still have intact neural pathways. If you put a camera in place of their eye and you hook it up, they should be able to see. Imagine if this is not speech recognition but speech production. You have a patient who's had a stroke, their left temporal lobe's been ablated, they can no longer speak, but you understand how they make sound. Could you move this to another part of the brain that's not involved in speech production, put a stimulator in, and basically replicate this in a different part of the brain? So this is all about neural rehabilitation and using machine-brain interfaces to produce sound. We already do this for motor. There's a number of people who are quadriplegic, who have no movement from the neck down. Using their brain signals, they can have a robot feed them chocolate. Okay? You guys, I'm sure you guys have seen similar stories to this. This is really hot. Eddie's just a superstar. How many neurosurgeons do you meet named Eddie, And he looks like he's about 12, but he's actually probably about 45 or 50. But this is like cutting edge neurorehabilitation. I think this is some of the coolest stuff I've ever seen. And then last is kind of a fun one. This is Ido Bachelet's work. George Church's lab just came out with another new paper the other day. Some people call it origami DNA. We call it nanorobots. Basically what these are nanobots. They're about 50 microns in diameter. They're made out of DNA. Basically it's a cage that has some sort of payload. And in this case it has 2 sensors. The newest one has 4. And basically what these are surgical robots. So you inject them in the bloodstream. They cruise around until their sensors find their target protein. So that could be CD19. It could be any surface protein you want. and then they unload. Okay? So in Ido's work, and this again was published in Science in about 2011 or so, his experimental subject of interest is a giant Israeli cockroach. These suckers are like this big. So he injects them with human tumor cells. He gets an established tumor. He injects the robot. They eradicate the tumors. And these things get excreted naturally because they're just DNA. So in 20 years' time, when you need to have your tumor excised, you won't go to the surgeon will come to you. Okay? So one last slide, and this is just gratuitous. I was fortunate enough a couple of years ago to have dinner in Cardiff with some really fun guys. So imagine having a dinner with these chaps. It was just the four of us. So this is Steve Dunnett. He used to be at the Brain Repair in Cambridge. He was one of the founders of the program in Lund on Parkinson's disease. Great guy, really, really fun guy. Chain smokes and flies airplanes at the same time. This is Martin Evans, who was, again, in Cambridge and then became chancellor in Cardiff. Martin is now 79. He has a cell therapy company. They have, I think, 4 programs in phase 2 where they can find different cell populations in tissues. They're very quiet about how they do this, but they've already shown safety and they have some really good efficacy data. This is Professor Sir John Gurdon, who won the Nobel together with Sheena in 2012. John is currently 87. Still active at the bench, still has funded grants, still publishes, works in the lab 3 days a week, spends 2 days running Cambridge colleges, will probably be at the bench until he's 120. The work he won the Nobel for, he did while he was a PhD student at Cambridge the year I was born, which was 1955. So John's really amazing, has his own institute in Cambridge called the Gurdon Institute. Really cool chap. I mean, he's an inspiration to all of us who want to continue to do science until we're a couple hundred years old. So finally, David, you'll be happy that that's it. As always, I don't know, Anders talks about 2 hours being a long time. But thank you very much.
Thank you very much.
I'm sorry, I thought it was only like 3:30. I didn't realize it was already.
So it's all fascinating. The masterclass. I don't think it's healthy for people to be sitting for too long. They say sitting is the new smoking. So I'm not going to have a long Q&A session, unfortunately, here. We should walk first.
Sure.
But I do want to ask, with all this treatments and possibilities, how long before we're going to cure dementia?
So
Are we going to still be having people with dementia and going terrible situations 50 years' time?
That's a really, really good question because, with all due respect to my longevity friends, we've been working, certain parts of my own research have been going on dementia for, 35, almost 40 years. We know nothing about dementing disorders. We know very, very little. And it's not surprising that 99.9% of the drug trials since the mid-1990s have failed. The idea that amyloid is the target for the disease is just stupid. Living to be 150.
So none of what you've highlighted here is likely to be correct, because I saw your charted applications of stem cells includes Alzheimer's.
Yeah, so that's not my slide, I have to say. I think stem cell therapy for Alzheimer's disease in terms of transplantation is never going to work. You'd have to transplant so much of the brain. I think stimulating the endogenous stem cell population in the way that was shown with stroke may have potential, but we need to understand more about what those cells do when they're activated, where they go, can we control them, do they create new neurons? If they all just turn into astrocytes, I don't think it's going to help. But the longevity community really irritates me because I've heard this from people from Singularity and others who say, oh, we're going to live to be 150. And then I say, okay, so what are we going to do about dementia? Oh, we'll have solved that in 10 years. I'm sorry, I don't believe that. I mean, if we did, we'd be developing potential drug therapies that show some efficacy. And everything, with the exception of cholinesterase inhibitors, has failed. And okay, so now people are starting to get a little bit smarter and say, well, maybe it's inflammation, maybe it's an inflammatory process. Okay, so how are you going to treat that, in a brain-specific way? I think dementia is a really, really significant problem, and it doesn't help when you have people like Pfizer and others who are just abandoning, work on neurodegenerative conditions. We need more money put on this, not less. But I think it's indicative of how big a problem it is.
So bearing in mind there's not much time, does anybody have a really burning and short question you want to chip in? Just come down here.
Thank you. Hi. It's going right back to the very beginning of your talk with regards to the Parkinson's
Yep.
disease and the transplants. You only transplanted in patient 4, you said, into one hemisphere
Yeah.
of the brain.
Yeah.
Would there be additional benefit to transplanting to both hemispheres?
Yeah,
Or
there would be, and most patients get bilateral transplants. He was so happy with his one transplant, he never wanted to have the other side done.
So what would be the physical differences with his progression and how?
Yeah, so his disease was somewhat asymmetrical. So he would be worse on one side than the other. And the only other thing to say is that the transplants do nothing for tremor. So if you have a really, really severe tremor, the transplants don't help. Deep brain stimulation may help. We have patients who, if you put a deep brain stimulator in their tremor, you can turn it off completely. But he had a very asymmetrical form of the disease. One side was essentially back to normal, and the other side just got worse and worse and worse. But he was happy.
Okay, thank you.
But yeah, normally you'd go for both sides.
Going back to the dementia and Alzheimer's issue, have you heard about the cerebral spinal fluid drainage theory that may cause things like Alzheimer's, where basically the brain is unable to sufficiently drain cerebral spinal fluid, and it can cause a buildup of various types of misfolded proteins and plaques?
Not so much that. I mean, I know that there are people who are investigating whether the kind of garbage disposal system in the brain is impaired, this thing we call autophagy, and maybe that the lysosomes aren't working quite appropriately. A lot of CNS disorders are alpha-synuclein disorders, amyloid disorders are caused by what appears to be mismetabolism or dysfunctional metabolism of these things. I've not heard it linked directly to CSF flow, but I have heard that people have looked in cells. So if you look in adult human aged brain, you see a lot of what's called lipofuscin, which is this proteinaceous material that just seems to be a byproduct of aging. If you look at patients with AD, they have a lot more of this stuff, but they also have a lot more amyloid. But you can see a lot of amyloid in patients who are cognitively normal. So there's something going wrong in aging per se. It's exacerbated in some way in AD.
Have you heard about cells trying to enhance lysosomes to break down lipofuscin?
Yeah, and I know other people are trying to do that as well. That's an approach that at least you're getting. I mean, the problem with the amyloid stuff, in particular the patients that have been treated, if you've already lost your hippocampus, you can clear all the amyloid in the brain you want. It's not going to bring back your cognition.
And to regenerate brain tissue as well.
Can you regenerate a hippocampus? I'm dubious about that. Yeah, that's definitely a beer conversation.
Maybe CA3 can regenerate itself, or we don't know. Anyway, I wanted to go back to Parkinson's.
Yep.
And one thing I don't really understand is you did show the progression of recovery for that particular patient after the graft,
Yeah.
which was actually building up in years.
He got better. Yeah.
Now that's confuses me, because all those original factors, whenever they are leading at the end of the day to oxidative stress, yada yada, destroying the cells.
Yeah.
Does it mean that they were very local and specific for the cells that died, and after they died they got eradicated somehow?
This was, I'll be very honest with you, the expectation was that, you might be able to get patients back up here, but then what you would expect to see is this, right? And that didn't happen. Now, it's clear that some of the patients who've come to autopsy, we find Lewy bodies in some of the transplanted cells. Because again, oops, I can go back to the previous slide. Again, the destruction is down here, okay? You don't see destruction up here. What you see is, neurite retraction and things like that. But all the inflammation and destruction is down here. So we're putting the new cells up here. So somehow we think, though it's, it's autopsy stuff, is that Lewy bodies are being transmitted up here and they're infecting, maybe in part what you were talking about. Somehow these are being transmitted into some of the fetal cells. But the vast majority of these cells are surviving a long, long time. And this is in a 67-year-old brain. This is not the most hospitable place to be putting brand new cells. And the fact that they've survived and not degenerated into disease is pretty surprising. Pretty surprising. And it may have to do that you're sequestering the cells away from the disease process. In AD or in Huntington's, it's going to be the opposite, because you're putting the cells immediately in a very nasty environment.
So, Stephen, you had to rush over some of the bluer sky parts. Are you interested in coming back at some stage and spending
Sure,
more time on the
sure.
bluer sky?
Yeah, yeah.
And it's all been fascinating.
Yeah.
We will have a chance to say thank you to Stephen in a moment. I'd just like to say we are moving to a pub, at least there's an option for anybody to come and join us. It's a different pub from where we've usually been, so we've got to experiment. The previous pub is great, but often is very noisy on Saturday afternoons, so people have said, can we find a quieter pub? So we're going to a different place. It's called the Royal National Hotel, which is in the opposite direction from usual. It's on Bedford Way, and there's a picture of that going to come up on the screen shortly. So if you want to continue some of this discussion, get a chance to talk to Professor Minger, chat to others in the audience. There's very knowledgeable people here. That's what I would recommend. Yes, that's the one. Slide number 7. Press F5.
Sorry. I'll let you do
The
it.
wonders of technology. There. So I encourage you to consider joining us there. There'll be other meetings in London Futurists advertised shortly. There's a few in the course of planning, but I think it's time to say thank you very much for a great masterclass of ideas, details, techniques, and a very powerful vision. Thank you very much, Professor Stephen Minger.